Okay, let's make a start. Thank you all for coming. I'll start with the acknowledgement to country. We acknowledge today that we're on the unceded land of the Wurundjeri people of the Kulin Nation, and we pay our respects to the elders past, present, and recognise that we have much to learn from our oldest living generations of scientists, teachers, artists and engineers who used to and cared for the land on which WEHI operates on, which is several sites, as you know. And before I introduce Haloom, I also wanted to just say something briefly about the connections to our Indigenous peoples and this particular disease. And you'll see on Haloom's first slide here 3 pictures. And these are pictures I took on a trip that I was privileged to go on last year in September up to the far north of Australia. And although Haloom won't be talking about that particular type of ataxia, we have the highest incidence of a particular type of ataxia in our Indigenous population, which is called SCAR3, spinocerebellar ataxia type 3, or Machado-Joseph's disease. And I was very privileged to meet some of the elders, and I had the privilege of sitting with one of the matriarchs at one of the sites we visited on Groot Island, and she took me through the pedigree of all the people that she knows of in these massive pedigrees, and we went through the pedigree on a piece of paper that was glued together and was 20 pages, and it was just one page after another, and there were like 50 people who were living or deceased who have this neurodegenerative disorder. So with that kind of entree, I now wanted to introduce Haloom. So Haloom has been a valuable and outstanding member of my lab since December 2017. She's a senior postdoc in my lab, and she obtained her PhD actually in epigenetics from the Baker Heart Institute in human epigenetic studies. So she has a wet lab background, and I think during her PhD she figured out that her future was not in the lab, but, well, in a dry lab, not the wet lab. So she went about sort of searching out a lab where she could change track, so to speak. So she thought about finding a lab, and I know now, she told me this, that she targeted my lab to come to, which I didn't know. So I was very grateful in the end that we ended up hiring Haloom 2 years out of her PhD. She quickly found her niche in my lab in repeat expansion disorders, which are a huge burden or a huge driver of risk loci in ataxias. And with her together, we have continued to build on our long-standing, over a decade-long collaboration with Professor Paul Lockhart's lab at the MCRI. And I know Paul is one of Haloom's biggest fans. So these days, she also works on repeat expansions in dementia and PD, and she just has a new preprint out on our PD work on repeat expansions. She won't be able to talk about that today. The talk is focused on ataxia. So Haloom's been an outstanding postdoc in my lab, and she has a fantastic track record. She's gathered multiple national awards. She currently holds an EL1. And she won the Lawn Millennium Award last year at the Lawn Genome Conference, and she also won the Australian Bioinformatics and Computational Society ECR Award last year as well. And she's a co-CI currently on an NHMIC IDEA grant, MJFF grant, and so on. So I hope you'll enjoy this seminar, which is really a sort of a tour de force through her work in ataxia, and it features some good old-fashioned and some nice new-fashioned genetic, statistical genetics and bioinformatics analysis. So please welcome Haloom. Thank you.
Good afternoon. Thank you, Melanie, for that kind introduction. So let's get into it. So I'll be focusing on the topic of hereditary cerebellar ataxia. And ataxia is a movement disorder that affects balance and coordination, which is not related to muscle weakness. It's caused primarily by impaired function of a region of the brain called the cerebellum, and this is primarily due to the dysfunction of a subset of cells known as Purkinje cells. Ataxia is a rare neurological disorder. It affects approximately 1 in 10,000 people, and although it is rare, it does still affect a lot of Australians, so approximately 3,000 Australians. live with ataxia. It is primarily an adult onset disorder, but it can also affect children. When we think about individuals who have ataxia and the different types of ataxia syndromes, we have two types of symptoms. There are the cerebellar symptoms, which typically occur with ataxia, so that may include the ataxia, which is the imbalance, as well as tremors and abnormal eye movements and impaired speech. But ataxia can also occur with non-cerebellar signs as more of a syndrome, and that's especially apparent in children. And these may include difficulty swallowing, vestibulopathies, neuropathies, and many other symptoms. So I just want to briefly talk about why genetic diagnosis is important for individuals with monogenic disorders such as ataxia. Even though people do have a clinical diagnosis of ataxia, without a genetic diagnosis and without knowing the specific type of ataxia, they are unable to access, for example, clinical trials that are targeted to a specific type of mutation, or obviously would be more successful than clinical trials just targeted to ataxia in general, and that's applicable for any type of rare disorder as well. It also gives answers to the family. It explains why this specific disease may be occurring in their family, and also alleviate guilt potentially, especially for childhood onset disorders where parents may think that it's something that they did that led to this happening in their child. It also means that people can join support groups for their specific disorder, and also is very important for family planning and allowing people to choose, for example, IVF to make sure they don't pass their condition on to their offspring. So I want to briefly introduce you to two members of our WEHI Ataxia Consumer Program, who I'm sharing with permission. This is 6-year-old Eliza, and her father is part of the WEHI Consumer Program. At 1 year old, she was diagnosed with a global developmental delay, and over time her symptoms became more severe and started to include ataxia, as well as hypertonia and delays to her fine and gross motor skills. At 2 years old, her parents accessed genetic testing, and she was diagnosed with a de novo variant in the gene CACNA1A. And these disorders are so rare that her paediatrician, who was not a neurologist, had never actually heard of this disorder. And initially, while this diagnosis was devastating for her parents, they finally had answers and her symptoms started to make sense, and they are now able to work towards better strategies to help her with her development. And importantly, her specific disorder does actually have clinical trials available for treatments at the moment that her parents may be able to access if that's something that they wanted. This is Jenny. Jenny has been part of our WEHI Consumer Program for, I think, about 5 years now. It's an absolute delight to meet with her every couple of months. She has an adult onset ataxia, and her father was initially diagnosed with this ataxia called spinous cerebellar 15, or SCAR15 for short. And at the time, this was many decades ago, there was no genetic testing for this disorder in Australia, and he had to be tested overseas. Jenny also opted for testing, and she was tested positive, and she now does have symptoms, and her sister chose not to be tested, and her adult daughter has also chosen to decline genetic testing. Her symptoms are getting progressively worse, and she has very restricted mobility, and unfortunately her father passed away from the condition last year. So when it comes to the genetics of cerebellar ataxia and rare diseases, we generally tend to think of point mutations and small indels causing disease, or possibly structural variants and copy number variants. However, ataxia is a little bit different to other rare diseases because the predominant type of genetic variant that causes ataxia are repeat expansions of short tandem repeats. You can still have point mutations and copy number variants, however, these make up the minority of ataxias. So I'll explain what we refer to when we're talking about short tandem repeats, or STRs for short. So short tandem repeats are repetitive elements of DNA that consist of motifs between 2 to 6 base pairs in length, so for example a CAG, and that's repeated over and over. We have more than 200,000 STRs defined in the human genome, and variation of these STRs is common and typically benign. The sites are very unstable, which is why we see a lot of variation between different individuals. However, for approximately 60 neurological disorders, these are caused by repeat expansions, so short tandem repeats that become expanded beyond a locus-specific threshold that can actually cause disease and become pathogenic. And what's interesting about repeat expansion disorders is that they're primarily neurological disorders. So, for example, ataxias, but also Huntington's disease, probably one of the more well-known repeat expansion disorders, and also frontal temporal dementias and ALS, as well as some epilepsies. So, pathogenic repeat expansions are very diverse. They can be inherited in biallelic or homozygous mechanisms, as well as heterozygous or X-linked. They can be in any part of the gene, so for example, introns and exons, and they can be many different types of motifs. And this is just a summary of the repeat expansions that have been discovered to date and where they're located in different genes. And you can see that many are located in exons, however, many are also located in introns. And I want to point out that you can see the majority of these are labelled SCA, which is spinocerebellar ataxia, again, one of the most common disorders caused by repeat expansions. So, one feature of repeat expansion disorders is that they are very heterogeneous. And I'll walk you through a couple of examples. So one example is a Fragile X syndrome, which many of you may have heard of, and that's typically caused by a repeat expansion in the gene FMR1 that is larger than 200 CGG repeats. However, there are also pre-mutations in this repeat expansion that are between 55 to 200 repeats that can cause different phenotypes that are less severe. So, for example, Fragile X-associated primary ovarian insufficiency or Fragile X-associated tremor ataxia syndrome. Another example is the CAG repeat in the gene ataxin-2. So typically a normal or non-pathogenic allele will be less than 30 repeats. However, what we found is that, and those that expanded larger than 33 repeats will cause a type of ataxia called SCAR2. However, some studies show that intermediate repeats, between 30 to 32 repeats in length, are actually a risk factor for ALS. And we've recently published a preprint, which also shows that repeat expansions that are larger than 33 repeats, but are interrupted with a different motif other than the CAG, so interrupted with a CAA. These do not cause ataxia, but instead are a risk factor for Parkinson's disease, and this has been shown in other studies as well. It's not published, but it is on medRxiv. And the final example is C9orf72 repeat expansion, which can cause either frontotemporal dementia or ALS, and we're not sure why it causes one rather than the other. Okay, one really important feature of repeat expansion disorders is the length of the repeat expansion itself. For most repeat expansions, the longer that repeat expansion is, the more severe the disease is and the earlier the age of onset. So this can lead to a lot of variation, even within families where one individual may have a pathogenic shorter allele and another individual may have a longer allele who then has a much earlier age of onset. Another really interesting feature is that we know that repeat expansions are very unstable, and they are inherited differently depending on whether it's maternal or paternal inheritance. So what we often find for many repeat expansions is when inherited paternally, they are actually contracted in the offspring, and when inherited maternally, they can actually become longer. So when it comes to genetic diagnosis of ataxia, it's an often long and difficult road, and many individuals don't actually access genetic testing and are just given a diagnosis of ataxia without knowing the specific genetic cause. One of the main reasons for this is that there are currently no high-throughput assays for repeat expansion diagnosis that are available in clinical diagnostic settings, and they still rely on PCR, and they only offer these PCR for the most common ataxias, and only the ones that were discovered quite a while back. The ones that have been discovered more recently and are also very common, there's currently no genetic testing. And for those individuals who do opt to receive a ataxia panel for the most common SDRs, only about 5 to 10% are actually diagnosed. And unfortunately, this test is no longer offered by VCGS as of about 6 months ago. So this is for individuals in Victoria, this test is no longer available, and they would need to reach out to interstate testing to receive a diagnosis. Another option is exome gene panels. However, these, until very recently, were not funded for adults by Medicare. So most individuals with ataxia never actually opt for an exome because they can be quite expensive. And in addition, they result in a diagnostic rate of less than 1%, given that they don't detect repeat expansions and they're only looking for point mutations, which are not a very common cause of ataxia. So that leaves us with a large number of individuals with ataxia who remain without a genetic diagnosis. However, in recent years, it's now become increasingly possible to detect repeat expansions in short-read sequencing data. So many diseases that are predominantly caused by point mutations have benefited from exome sequencing that's readily available for many different disorders. However, as I mentioned earlier, that has not been the case for ataxia because they're primarily caused by repeat expansions, and historically, repeat expansions are very difficult to detect in short-read sequencing data. However, over the last 5 or 7 years or so, it's now become increasingly feasible to detect repeat expansions in whole genome sequencing, especially paired-end sequencing data. So I'll walk you through a couple of examples. For STRs that are shorter than the read length, as shown in this example here, we can determine the exact size of the repeat because it's located entirely within a read. For STRs that are longer than the read length, which is typically 150 base pairs, we can't get the exact size, but we can get a pretty good estimate depending on the locus. So for some loci, we get a much better estimate than others. And sometimes we just need to know that there is an expansion, and that's enough to say that this person can be diagnosed with that specific disorder, even if the sizing isn't accurate. Some STRs can be quite complex, and what we find is that there's a pathogenic motif shown in red that is side by side with a benign motif. That also makes detection of the STR and sizing inaccurate, but sometimes, again, that is sufficient for diagnosis. And this is one example where short-read sequencing falls short, where the pathogenic motif is embedded deep within a benign motif, as shown here, and because of the short nature of short-read sequencing, we actually can't get uniquely mapping reads for that pathogenic STR, so we may actually miss that diagnosis. However, in general, there's a lot of papers that have been published, including from our group, that show that short-read whole genome sequencing is very feasible for diagnosis and detection of short tandem repeats and their expansions. So this is a timeline of the discovery of repeat expansions over the last 30 years or so. So you can see that in the early days of discovery, there was a very rapid discovery of repeat expansion disorders, and this was primarily using methods such as linkage analysis and molecular methods, and especially for repeat expansions that are located in exons. However, around 2003, the discovery really started to slow down, and it didn't pick up again until just around 2019, when we had a lot of these advances in bioinformatic tools and also long-read sequencing for the detection of repeat expansions. So now there has been an explosion of repeat expansions that have been discovered in the last 5 or so years. Okay, so I just want to give a brief overview of what I'll be talking about in terms of our research for this presentation. I'll start with how we developed our ataxia research program and the discoveries that we've made, and I'll then talk about how we can work towards improving genetic diagnosis and discovery of ataxia genes, and then finally focus on what are the next steps for discovery and how we can improve discovery. So our story of ataxia actually started quite a while ago, before I joined Melanie's lab, and it started with a disorder known as CANVAS. So CANVAS stands for cerebellar ataxia with neuropathy and vestibular areflexia syndrome. So to break that down, that's a syndrome that includes people who have cerebellar ataxia, as well as neuropathy, which is loss of sensation in the hands and feet. It also has this vestibular areflexia, which is impaired function of the ear for regulating balance. And for some individuals, it also includes a chronic cough. So this disorder was actually described primarily by a Melbourne neurologist called Dr. David Schmulowitz, and he was the one who really pioneered our understanding of this syndrome and described the symptoms that occur with ataxia that make this a unique syndrome. And the reason why CANVAS is so unique is that the majority of ataxia disorders don't have any distinct non-cerebellar symptoms. However, CANVAS does, and for that reason, it was one of the few disorders where you could diagnose people with CANVAS without actually knowing their genetic the genetic cause of their disease. And what's also really interesting is that it appears to be a recessive inheritance pattern. So many families had siblings who were affected, but the parents were not affected, indicating a recessive inheritance pattern. And there was also quite a lot of families who presented to David with canvas. So it appeared to be fairly common. So the study started, I think it was around 2011, 2012, so quite a while ago. And it was a collaboration between Dr. David Schmulwitz, who is the lead neurologist who understands canvas, as well as Professor Paul Lockhart from the MCRI, who is a molecular biologist and a long-term collaborator with Melanie. So it started with 22 families who had suspected canvas. And the reason I say suspected is that without a genetic diagnosis, we can't know for sure that they're canvas. But David did a pretty good job at phenotyping, so it does appear that the majority of them would have canvas. And at the time, whole exome sequencing was performed on 23 individuals from 15 families. However, no shared rare or de novo variants were identified in any of the families. So it remained unsolved. However, as I mentioned, the canvas runs in families. So what we were able to do is run linkage analysis on these families. So this is an example of one of the large families with canvas, which we've called Canvas 9. And this family had 4 affected siblings. So what we were able to do is to basically compare the DNA of the affected siblings to the unaffected siblings and look for regions that are shared in a recessive method in the affected siblings that are not present in the unaffected siblings. And what we found was a very strong peak on chromosome 4, indicating that there is potentially a genetic cause located in this region. And given that we had so many families, we were also able to run linkage analysis on other families as well. And when we compared the results, we found that there was a small region on chromosome 4 that was inherited in a recessive fashion in all 5 of these families. So we were very confident that there is a genetic cause that could be located on chromosome 4. So at this point, knowing what we know about ataxias primarily being caused by repeat expansions and that many of them are intronic, we realised that whole exome sequencing probably won't cut it. So we then moved to doing whole genome sequencing for 2 unrelated individuals. So one person from the family Canvas 1 and one from Canvas 9. And at this time, Melanie was developing a tool with a team at Illumina called Expansion Hunter De Novo. And at this time, the tool had not yet been published, but because I worked with Melanie, I had access to the tool before publication. And this is important because Expansion Hunter De Novo was the first tool that was able to be a catalogue-free screen, and basically unbiased screen of whole genome sequencing data where you don't need to know the existence of an STR beforehand to be able to identify the STR in your cohort. So we had 2 individuals with canvas and 31 controls. And when I ran Expansion Hunter De Novo, one of the top hits was a repeat expansion that was inside the linkage region, and it was an AAGGG motif located in the intron of the gene RFC1. And this was present in both individuals who have canvas, and it wasn't present in any of our controls. So this is what the region looks like if we visualize the region in a genome browser, so in IGV. This is what it would look like in a control. In the reference genome, there is a STR present here, but it's an AAAG motif. What we find when we look at our 2 individuals who have canvas is that that reference motif is not present, and instead they have 2 copies of this AAGGG expansion that replaces the reference STR. However, this is still only 2 individuals, so not really enough evidence to say, let's go and validate this in the lab. However, we did have whole exome sequencing. Now, whole exome sequencing wouldn't be expected to capture this repeat expansion because it's located deep within an intron, but fortunately, WES protocols can be somewhat dirty, and sometimes there are intronic reads that are present. So we manually looked at individuals who had whole exome sequencing data, and we checked to see if they had any evidence, even just one read, of the AAGGG motif rather than the reference motif. And you can see in this example, this individual does have some evidence of that motif, and this individual does not. So what we found was that out of the 14 individuals who had at least one read that spanned across this region of the genome, 11 had the AAGGG motif. And keeping in mind this motif is rare in controls. So that's pretty convincing evidence for us to go to our molecular collaborators and say, we think this is it and we need to validate with PCR. So this is our cohort at the time. All these individuals had a clinical diagnosis of canvas, and when our collaborators did the repeat prime PCR, the vast majority of them were confirmed to be biallelic for the AAGGG expansion, indicating that this is the cause for canvas. However, 4 individuals did not have the repeat expansion. So what we did, we then looked more closely at their DNA. We performed whole genome sequencing for those individuals, and we were able to diagnose 3 of them with a different disorder. So, for example, one of them had SCAR3, and the other 2 had, again, SACS for one and SCAR45 for the other, and we were very confident with that diagnosis, and only 1 remained without a diagnosis. So at this point, we were quite confident that this is the cause of canvas. So just to summarise what it looks like, this is an example of what canvas looks like in the reference genome. There's a non-pathogenic allele with the AAAAG expansion. With the AAAAG STR, and in individuals who have canvas, they have 2 copies of the AAGGG pathogenic allele. And if we look more closely at the DNA that surrounds this repeat expansion in the individuals who have canvas, we notice that they have a shared haplotype. So what that indicates is that this repeat expansion doesn't occur spontaneously. It occurred once in history, probably a very long time ago, and all these individuals inherited this repeat expansion from a common ancestor. And if we look at the region in the UCSC genome browser, highlighted here in blue, that's the common region shared by the individuals with canvas. You can see there's very strong linkage to disequilibrium at this region. And we were able to use an algorithm to determine that based on the extent of DNA sharing, the most recent common ancestor where the mutation most likely arose lived approximately 25,000 years ago. So we used the app that I developed based on a method published by Melanie that allows you to determine the age of a genetic variant. So we published this work in 2019, almost 10 years after the project first started. And at the same time, there was another paper that came out from the UK who also made the same discovery. And this team put a lot of work into trying to identify the mechanism that causes canvas. And they really couldn't find anything, and we still don't know the exact mechanism that canvas arises. So how does the repeat expansion cause disease? That's still somewhat of an unknown. And since then, canvas has been diagnosed in countries across the world and across most continents. And we now know that it's one of the most common causes of ataxia worldwide. And just one interesting finding I want to highlight. For some individuals with canvas, specifically from the Asia Pacific, they actually have a different motif that causes canvas. And this is something that's quite unique that hasn't really been shown for other types of repeat expansion disorders. They have an ACAGG motif that causes canvas with very similar phenotype to the main motif. And after we published our paper, there was a lot of interest in the tool that we used, which was Expansion Hunter de novo. Oh, sorry, I just want to actually point out, how did this team discover this repeat expansion if they didn't have access to Expansion Hunter de novo? So given that we had very tight linkage region and this team had a lot of families, so they knew exactly the gene that they were looking at. So someone basically sat at the computer and scrolled across the gene RFC1 in the IGV browser and just looked at the DNA and just saw that there was something there. So they had to do that visually. Obviously, you can't really do that if you don't know the exact gene that's causing the disease, which is why there was so much interest in our method, which was Expansion Hunter de novo. So Canvas was also highlighted in this paper as a successful application of the tool, and this is now one of the most widely used tools for the discovery of repeat expansions. Okay, so the success of our Canvas story led us to develop an ataxia research program, which we called GREAT. So that stands for the Genetic Research on Repeat Expansions in Ataxia. So at the time, we had about 100 adults. So this is about 2020. We had about 100 adults recruited into the program, and they were recruited by our neurologists who did a very thorough clinical assessment. And we also have very strong collaboration, again, with the team at MCRI, so especially Paul Lockhart and Penny Snell, who is the coordinator, and Kaylee Davis, who is a PhD student who works on this project. And without this collaboration, we would definitely not be where we are today. So it's a very fruitful and a very important collaboration for us. And again, highlights how important it is for people from different aspects of science to come together for this type of research. So the broad aims for the bioinformatics aspect of this program was firstly to diagnose people with known ataxia genetic disorders, and for those who remain without a genetic diagnosis, we want to see if we can discover more novel ataxia genes. So I want to highlight one of our findings. So we had an unbiased screening of a small ataxia cohort, and we were looking for novel repeat expansions. So at the time, we only had whole genome sequencing for 47 unrelated ataxia cases and about 200 controls. And this is going to be a very different story to the Canvas discovery, because we don't have a defined syndrome. We have a mixed adult onset ataxia cohort, and these people are expected to have different genetic disorders that causes their ataxia. There's no predefined syndromes in this cohort, there's no family linkage studies, and there's no known inheritance patterns. So basically all we know is that they have an ataxia, and the neurologist has decided this is most likely a genetic cause and not an acquired ataxia. So we once again analysed the whole cohort with Expansion Hunter de novo, and we noticed a repeat expansion that really stood out to us. So this was an intronic GAA expansion in the gene FGF14, and it was present and expanded in multiple cases compared to the controls. And the reason why it really stood out to us is that firstly, FGF14 is a known ataxia gene. Point mutations in this gene cause a disorder known as SCAR27. However, it is a very rare disorder. But also, there's precedent for the pathogenicity of the GAA motif. There are many repeat expansion disorders that are caused by that motif. So collectively, based on this information, we thought this looks like this could be a real repeat expansion that causes disease. So we went back to our molecular biologist collaborators, and they then did PCR sizing of the FGF14 GAA repeat expansion in the cases and the controls. And we also had additional cases that had not yet been sequenced, as well as additional controls. So the cohort for this analysis was 95 cases and 311 controls. So with the PCR, we're able to get a very accurate size of the repeat expansion. And what we found was that for repeats that are larger than 335 GAA repeats, there were 4 individuals from the ataxia cohort who had a repeat of that size, but no controls. And we call this the fully penetrant range. However, we also had this incomplete penetrance range, where there's a strong enrichment for individuals in the ataxia cohort who have expansions that are larger than 250 repeats compared to controls. So we had about 9% of the cohort were actually in this range in the ataxia cohort compared to only 0.6%. So that indicates that this range can also cause disease, but not necessarily always. As we can see, there are 2 people in the controls who have this disease. So that's why it's called incompletely penetrant. And we then reached out to our German collaborators, this work was led by Dr. Katja Lohmann, and they found that this repeat expansion was present in 8.7% of their ataxia cohort in the full penetrant range. So at this point, we were very confident that we had just discovered a repeat expansion disorder that causes adult onset ataxia. That's actually a very common cause, and no one had actually known that this disorder existed because it wasn't a previously defined syndrome. And just briefly, we also looked at the correlation between the length of the repeat expansion and the age of onset. I mentioned earlier that longer repeat expansions can cause a reduced age of onset and more severe disease. So what we found when we did this analysis on the combined Australian and German cohorts was that for every 100 GAA repeats increase, there is a reduced age of onset by 11 years. And I just want to point out that this isn't it is a strong correlation. However, there's a lot of variance in the data that's not explained only by the length of the repeat expansion, and I'll talk about what could be potentially leading to this variation later on in my talk. So we published this work in 2023, and once again, at the same time, another team also made the same discovery. So this time it was an independent discovery in a French-Canadian cohort, and what was very interesting about this study is that in an undiagnosed adult onset cohort of ataxia, they discovered this repeat expansion present in 60% of individuals who have ataxia. So this is obviously a very significant cause of ataxia in this community, and it indicates a founder event in that population. And they also had collaborators in Germany, Australia, and India, and they also show that there are individuals from those countries who also have FGF14, but also not as high as 60%. So one of the really interesting features of this study is because they had so many families with this disorder, they were able to look at the paternal and maternal inheritance of the repeat expansion, and what they show is that when the expanded allele is inherited from the father, it's contracted, and when it's inherited from the mother, it's expanded. So over many generations, you can imagine that there's this yo-yoing of the length of the repeat expansion, and if it falls below the pathogenic threshold, then you have people who have the same locus who inherit, so individuals who are inheriting the pathogenic allele from the dad, but it's not actually pathogenic because it's below the threshold. So that can make repeat expansion discoveries quite difficult. Based on our work and the work of the Pellerin team, SCAR27 has now been updated in OMIM. So the original SCAR27 is now SCAR27A, and the repeat expansion disorder is now known as SCAR27B. And once again, in the time since this work was published, it's made a worldwide impact, and people with this disorder have been diagnosed in many countries across the world. And what's really interesting is there is now a potential treatment for SCAR27B. So for AP is a drug that has been approved for multiple sclerosis and has previously been suggested as a possible treatment for ataxias, and there is some evidence to suggest that it may actually be helpful for people with SCAR27B. There's also SCAR27 Ataxia Foundation that was established, so a support group for people with the disorder, and one of their main advocates is someone you may know, so Bill Nye, the science guy. And the reason he's the advocate is because he actually has ataxia that runs in his family, so he doesn't personally have it, but his siblings have it, and it turns out they have SCAR27B, and it's been in their family for centuries, and they didn't really know what it was until the gene for SCAR27B was discovered, and they were able to receive a genetic diagnosis. And if you're interested, he has a lot of videos on YouTube about his family story. Okay, so just to summarize the first part of my talk, we've now established an ataxia research program, and we've made 2 discoveries of genes that cause ataxia, that cause canvas and SCAR27B, which have turned out to be 2 of the most common ataxias worldwide. And we are now funded to recruit an additional 800 ataxia cases to continue with our studies. So just briefly, the next part of my talk will be focusing on how we can improve genetic diagnosis and also improve discovery of ataxia genes. So we recently published a paper, it's just come out this year in Genome Research, where we focused on how to improve genetic diagnosis of people with ataxia, given that most people have only had an ataxia PCR panel before. So we recruited 110 individuals with ataxia who had a negative SCAR panel. So basically they were tested for SCAR 1, 2, 3, 6, and 7, and they remain without a genetic diagnosis. And we did short-read genome sequencing on all these individuals, and we focused on trying to diagnose their ataxia. So we ran our repeat expansion detection pipeline, and so we ran that in our lab, and our collaborators have access to a number of tools that are just more like websites where they can look for small variants such as SNPs or copy number variants or mitochondrial variants. So they looked for variants in known ataxia genes that may be causing ataxia in these individuals. We also did long-read adaptive sequencing on these individuals, just because of the nature of repeat expansions, they're very long. So logically, long-read sequencing would be a logical way to be able to detect and accurately size repeat expansions. However, long-read sequencing, if you want to do whole genome sequencing, is very expensive. So we opted to do adaptive sequencing with nanopore. So we basically define the regions around the repeat expansions we're interested in, and the pore will only sequence DNA that comes from those regions. So that's a much more affordable and easier way to be able to potentially size repeat expansions. So we wanted to see how this compared to short-read sequencing data. And the main reason for that is that short-read genome sequencing is it will be very easy to implement clinically as a potential clinical diagnosis, and we wanted to see how that would compare to long-read sequencing. So to summarize our results, we identified 33 pathogenic repeat expansions from the short-read sequencing data, and an additional 9 that we considered to be variants of unknown significance. And we also identified 7 small variants that were likely pathogenic or definitely pathogenic in 7 individuals, and 9 that are variants of unknown significance. So that variant of unknown significance basically means it's likely to cause the disease, but we don't have enough evidence to say this variant is actually going to affect the function of the gene. When we compare to the long-read adaptive sequencing, we detected 29 repeat expansions. And the reason why we missed out on those additional 4 is just due to the nature of the long-read adaptive sequencing. A lot of samples had low coverage, so some of the repeats we actually weren't able to detect due to that poor coverage issue. So overall, we solved 36% of this unsolved cohort, which is a very large proportion, and as well as 16% having a variant of unknown significance. And the majority of these diagnoses were repeat expansions, so 24 of them were SCAR27B. 5 were Canvas, but I want to point out that we have a parallel Canvas research program, so most of our individuals who were suspected of Canvas went into that program. If they had gone into this study instead, we would have probably had a much higher success rate as well. And we also had 2 SCAR8s, 1 Friedreich's ataxia and 1 SCAR36. So our main take-home message from this study is that you really can use short-read genome sequencing for diagnosis, and you get a really high diagnostic solve rate. So from our point of view, there's no reason why this shouldn't be implemented in a clinical genetics testing setting where they're already doing whole genome sequencing for other disorders. Okay, so I want to talk about one other study that was published a couple of years ago now, which was a collaboration with a French ataxia team. So this was a discovery of point mutations in the gene PMPT1, which cause a type of ataxia called SCAR25. So what's interesting about this study is that this French family was originally published in 2004, and they had a really strong linkage region on chromosome 2. And this is our Australian family, who we analysed back in 2015 and also discovered a PMPT1 variant. However, the problem was that this gene was already associated with a different recessive disorder, which didn't really make sense because our family was, had a dominant inheritance pattern, and also it was the wrong disease. So we overlooked that variant for a long time, and I spent a lot of time analysing for repeat expansion CNVs, other types of variants in this family, until the French team reached out to us a couple of years ago and they said, hey, we found a variant in PMPT1 in this family. Did you have variants in that gene as well? And yes, we did. So once they told us that, we realised actually that PMPT1 was the variant causing the disease in this case. So the key take-home message from this is that there are a lot of factors that can complicate the discovery, even when the variant is right there in front of you. So disease pleiotropy, where a single gene can cause different disorders. Also, variant interpretation can be difficult, and it also highlights the importance of collaboration. So we didn't know it was the right variant until the other team reached out to us and said they had the same variants in the same disorder. So since that paper was published and our cohort has grown, what we've actually realised, as we've had more individuals with ataxia, is that we found people in our cohort who had the exact same splicing variant in PMPT1 as our original family, and these people were not apparently related to that original family. So we suspected that what's actually happening is these people are probably very distant relatives. So by looking at the DNA around the PNPT1 gene in these individuals, we're able to identify something called identity by descent, which means that region of DNA was inherited from a common ancestor in these individuals. And we show that they are actually related, and they're about 10th degree relatives. So eventually we would have realised that the PNPT1 variant was the causative variant, even if that the French team didn't reach out to us. However, the important take-home message from here is that we can screen our cohort for regions of identity by descent without knowing what the pathogenic variant is, and we can identify clusters of individuals who share regions of identity by descent and then link them up into mini families, and then that gives us a region of the genome where we can say there's likely to be a pathogenic variant in this region that's shared by these individuals. And that's one of the methods that we're implementing now in our cohort, especially as our cohort is growing and this is becoming more feasible. And we've already found in total 3 clusters like this where they do share a known variant. Okay, so to summarise part 2, we have improved genetic diagnosis in unsolved ataxia cases by approximately 36%, and we're also establishing new methods in our lab for detection of novel discoveries. So the final part of my talk will focus on what we can do in the next step to facilitate novel ataxia gene discovery. So I want to talk about this really interesting study that came out last year looking at somatic expansions in FGF14. So what this team did, so this is the other team who discovered SCAR27B. So they took post-mortem brain from individuals who have SCAR27B, and they sized the repeat expansion in different regions of the brain, including the cerebellum and the cerebellar vermis. And what they found, this is an example, they found that the repeat expansion is actually larger in the cerebellum and the cerebellar vermis compared to other regions of the brain. So other regions of the brain have the same size as you would expect to see in blood. So in this case, it was 288 repeats. But in the cerebellum, it's actually much larger than 300 repeats. So this is a phenomenon that has been observed before, especially in Huntington's disease, and is very well characterized. So what's been found in Huntington's disease, which is a severe repeat expansion disorder and a very devastating disease, is that even though people start off with a repeat expansion length of maybe 30, 40, 50 repeats in length, what happens over time as the disease progresses is that you see repeat expansions in a subset of cells called the SPN cells, which are the cells that primarily are affected by Huntington's disease. And the expansions of those CAGs are no longer 40 repeats in length. They're now up to 500 repeats in length. And this is when you have that loss of neurons and that end of life. So what that indicates is you may start off with a repeat expansion of whatever length in your germline, but the development of the disease depends on that somatic expansion in a specific cell type, which is what causes that end-of-life event. So when we study the age of onset in Huntington's disease, we find that there are SNPs that are associated with age of onset independent of the length of the Huntington's disease repeat expansion. So this is an example of a GWAS where they have 9,000 individuals. They all have Huntington's disease, and the phenotype they're testing is the age of onset, independent of the length of the expansion. And they find many different SNPs that are associated that regulate this age of onset. And these are primarily genes that regulate DNA repair mechanisms. So the main mechanism is the MSH3 DNA repair pathway. And the biology behind this is that MSH3 forms a complex which is required to fix these loops. So in the context of Huntington's disease, you have a CAG repeat loop here, which occurs during DNA replication, and that needs to be fixed before DNA replication can continue. And the way it fixes it is not by cutting it out, but rather making an excision on the other strand and then inserting a repeat expansion in this allele as well. So this is how you have that increase in the repeat expansion length. So what has been shown in Huntington's disease is that these SNPs in DNA repair pathways result in this it increases the speed in which these somatic expansions occur in the brain, and as a result, can lead to reduced age of onset and increased severity of repeat expansion disorders. So what this means is that repeat expansion disorders are not purely monogenic. There are actually polygenic factors that can regulate disease onset and progression. And this is likely what's happening here in SCAR27B, and likely to be the reason why we see variance in this figure that's not explained by the length of the repeat expansion alone. However, no one's really looked at the genetics of the polygenic contribution of DNA repair pathway genes to SCAR27B, and that's something that we're really interested in doing further research on. So what's next for our program? We are now funded for an additional 800 whole genome sequencing for ataxia cases, and we are recruiting from across Australia. We have a lot of interstate collaborations, and we also are recruiting from New Zealand, and we may not be able to achieve the full 800 from Australia and New Zealand alone, so we've reached out to collaborators in the US who may potentially contribute samples as well. We will still be doing short read sequencing. However, we also will be moving into long read sequencing for cases who remain unsolved to see if we can improve that diagnosis and detection of repeat expansions in long read sequencing. Our focus will be again on novel gene discovery, and I think with CANVAS and SCAR27B, they both have factors that made the discovery a bit easier. CANVAS was a known syndrome, SCAR27B was a known ataxia gene, and both were very common. I don't think it will be as easy going forward, so we'll be developing new methods to facilitate this discovery. We also have a very strong interest in this interaction of monogenic and polygenic factors in ataxia, which we'll be working on further, and that will be facilitated by our accessing large biobanks. So the UK Biobank and Genomics England, for example, have a combined 1,400 ataxia cases who we'll be also including in this analysis. Okay, so thank you, and I just want to acknowledge my collaborators who were key for this work. Of course, my lab head, Melanie Barlow, and Paul Lockhart from MCRI, as well as Kaylee Davis and Penny Snell, and of course, Dr. David Schulwitz. Thank you.
Hopefully you can hear me. Yep. Okay, questions? Yeah, I can see Marnie's already got her hand up.
Thanks, Haloom. That was really interesting. I might have missed it. Apologies if I did. So for CANVAS, when they've got their their 5 bases are actually different from controls, I guess. Is that repeat also expanded? So is it expanded and changed? And what's the known about that pathogenic mechanism? I know that's not what you work on, but how does it relate all in together to give you the disease?
Yes, so it is an expanded AAGGG, and it's very large. I think it was like 2,000 base pairs. However, we actually don't have a threshold. So there are people who have CANVAS who have alleles as low as 200 repeats who still have CANVAS, and there hasn't been a lower threshold yet. So it's a bit strange. So it doesn't seem that you need to have a very large expansion to get CANVAS as long as that motif is present. And in contrast, you can have 2 expansions of the reference motif and you don't get disease. So it's really dependent on that specific motif. And we still don't know what the mechanism is for CANVAS. There's been studies looking at the DNA structure that is affected by the expansion, but I don't think that's very solid yet. They've looked at gene expression, they've looked at protein function, they haven't found anything. So we still don't know.
Fascinating work, congratulations. I didn't know much about repeat expansion disorders before today, but I think I'm missing something. Why do a series of common molecular problems, i.e. repeat expansions, in a bunch of different genes all cause the same phenotype? I think I'm missing something there. Are they all a common family of genes?
Well, we're all missing something. We still don't know. So as I said, the repeat expansions all cause neurological disorders, and it's different types, but most of them are ataxia. So why do so many different repeats in different genes that don't have overlapping function or cause ataxia? So we don't know. It could be that those genes are highly expressed in Purkinje cells, and it may be that it's only in the Purkinje cells in the cerebellum where you have that somatic expansion happening, which may be contributing to ataxia, in contrast to Huntington's disease, where that somatic expansion only occurs in a specific subset of cells in the cortex. So there does seem to be some cell type specificity that leads to the different diseases, but we actually don't know.
But are there, I guess you did show the data, but how many ataxias are caused not by repeat expansions?
Okay, so there are hundreds of genes that cause ataxia that are not repeat expansions, but they're individually rare. And also they're not pure ataxias. Most of them are not pure ataxias. Most of them are part of a more severe syndrome that has a lot of other symptoms. So it's a bit complicated. But in terms of numbers of people who have ataxia caused by repeat expansions, it's predominantly repeat expansions, but there are hundreds of other genes. Okay. And if you want to think about families of genes, many of them are DNA repair pathway genes, but not all of them. So really, we still don't know.
These are the non-repeat genes you're talking about, or the repeat?
Both. So for example, RFC1 is part of DNA repair pathway.
Okay, but it's still weird, right?
It is weird. We just don't know.
So weird.
Can I follow up? That was super fascinating. Thank you very much. So sort of following on from this, what you showed in the last couple of slides is that if these ataxias are now not monogenic diseases, but polygenic diseases, and they have a specific phenotype where suddenly you have just in parts of your brain, you basically generate these poly-repeat expansions as an explosion in this particular, can you still say what you just said? That, some of these ataxia genes are not due to a repeat expansion? Because if they just explode in your brain in a particular type of neurons, you know what I mean? So does it make sense?
Do you mean can genes that cause ataxia that are not repeat expansions may also be repeat expansions?
That you can't identify as repeat expansion genes in a whole genome sequencing, but
right.
might become repeat expansion genes in somatic.
Yes. So it does appear that you need to have a certain threshold in the germline first before you become susceptible to that expansion. Okay. So for example, in Huntington's disease, if the threshold is 33 or 35 repeats. If you have 30 repeats, you don't see that somatic expansion in the brain, so you don't get Huntington's. So you have to have at least 33 repeats, that's the baseline, and then it becomes expanded. It's because the length of the STR means it's now become inherently unstable, whereas the shorter alleles, they're very stable, so they don't have that somatic expansion. But it's possible there may actually be cases where we can't really figure out, like, if that threshold may be only one base pair longer than the reference, for example, how would we pick that up in the germline? So potentially that may be an issue. But we can't really access brain that easily either.
Can you use the genes that are SNP affected, your polygenic genes, can you use that as a pre-screening to potentially see whether whether there's other
right, so individuals with an unsolved ataxia who have a high burden of those SNPs. That's an interesting idea. I think that's something we haven't really thought about. But so that's kind of looking at polygenic risk scores. And in our experience, there's a large overlap between cases and controls in terms of polygenic risk score. So you really see the distribution shifting in a large population rather than individuals. But it's still something we can look at.
I think David's question is a really interesting one. I don't think you can screen, but we can.
Thank you.